Welcome to our virtual space, where thought leaders who, in a variety of ways, have committed themselves to improving our lives share their work, perspectives on current affairs, and what brought them to where they are today. My name is Rob Liu, and this is the Xchange. So, Ramona, thank you so much for joining us in this sort of portion of the Xchange. As you know, what the Xchange tries to focus on are individuals that are working in science, as well as a broad range of fields that connect with, in particular, in our first series, how COVID-19 has changed our lives and even changed how we work. So, one thing that we'd really like to do, though, is sort of set the stage for our listeners and our viewers by hearing from you sort of the kind of work that you do, but also what brought you to science and to your particular focus on infectious disease in South Africa.
Thanks for having me. It's nice to be part of this initiative. So just to talk a bit about what it is I do. So I work on an infectious disease, but a human parasite, which is the disease Leishmaniasis. And this disease, although not endemic in South Africa, is endemic in other parts of Africa, Asia, South America, and India. And this disease is transmitted by a vector, a sandfly. So very similar to the malaria parasite, which is transmitted by a mosquito. But what makes this disease worse is that whereas malaria gives you one form of the disease, Leishmaniasis gives you three. So it's not just morbid, it's also lethal. And we don't have a vaccine, and our drug therapy is suboptimal. What drew me to it was that Leishmania research in South Africa was minimal. When I joined the team, I was the only female that was working on it, and I still am in South Africa. And what got me to science as a whole was during my master's degree. I'd say rather during my honors degree and molecular biology and things like that. I started off in undergrad learning about marine biology as well as plant biology and molecular biology. But molecular biology fascinated me, how little vectors and little genes can be inserted and result in growing things or making things like a protein or growing a plant in a test tube. So that is what fascinated me. And then towards the end of my honors degree, I started learning more and more about infectious diseases, reading more about it. And I started off working with malaria. And then it just introduced me to this whole world of diseases where malaria was just one of them. And Africa was afflicted with so many. So after my master's, I decided that I wanted to challenge myself and I wanted to go further into the diseases. And I decided upon Leishmania because so little was being done on it at the time.
So, what I've heard and what I've read is that in some ways, Leishmania is considered sort of a neglected disease.
Absolutely.
In the sense that yes, can you tell us a little bit more about why it's sometimes classified as being neglected?
Very much so. Neglected mainly because very little research is being done on it. And second, very little research money is being poured into it. If you look at the other diseases such as HIV, TB, and malaria, it gets the majority of the funding when it comes to this disease. So even though neglected diseases as a whole afflict more of the world's population overall than either of these three diseases, so not only that, it's also the underreporting because of the remote areas that the disease might be found in, the lack of proper healthcare and infrastructure to report on the disease. And that's part of the reasons that it's neglected, yes.
So I know that one of the things that interests you is the way in which models can be used to understand a human disease. Can you share a little bit more with us on what models are you using or focusing on? Why are model organisms so important?
So in our case, like I just mentioned, there's no Leishmania disease in South Africa. So getting patient samples and being able to work with biological fluid, etc., was really difficult because we don't have it. And bringing it into the country is also difficult because of the Department of Health, etc. If we don't have the disease, they're more concerned to bring it in, even if it's just a sample. So we had to develop some other way for us to study the disease, and hence the biological organism came into play. And the one that I'm interested in using is a mouse model of disease. So the mouse model of Leishmaniasis was established around the 90s. And I think the reason the mouse model specifically was that at the same time, gene deletions and transgenic mice and things like that were also coming into play. So it was possible to knock off certain genes that you thought was important in the disease and look at it as a whole. And because of the similarity between the mouse mammalian immunity and human immune system. So we started using the mouse model to initiate the disease, and we were successful to do that with both two forms of the disease being cutaneous and the lethal form being visceral. Apart from that, in the 90s, it was great to use mouse models because you were already getting so much of information. But by the 20th century, there's a big drive for translation, where you need to translate the studies that you get in a mouse model to human. So at this point, we also have now started using human cell lines, peripheral blood mononucleosides, to sort of more represent the human system and translate what we see in the mouse system. Is the same thing happening in a mammalian cell line, for example.
So I know that one of your particular areas of interest intersects with the immune system,
Yes.
with immunology. And I don't know how it is in South Africa, but in the United States, of late, for the last several years, there's so much direct advertising by drug companies to patients, to the general population. And what is striking, and students ask me about this all the time, is that they notice that if you look at many of the drugs, they have some interaction with the immune system. They modulate the immune system.
Absolutely.
So clearly, the immune system has become such an important focus in terms of understanding human
Absolutely.
disease. Yeah.
Absolutely. Most pathogens have devised ways to dampen the immune system so that they can initiate infection and multiply. So whereas the drug will be able to be antimicrobial and kill the bug, the immune system might still be struggling. So you need a combined effort between the two. And that's the type of work we actually do, where we try to find host-directed therapies. So basically host factors, host immune pathways, host molecules that can supplement a drug as an adjunct therapy to get a better clinical outcome.
Yes, fantastic. So we're all globally going through this COVID-19 pandemic right now. And of course, there is the challenge that I'm sure you're faced, which is for doing research. Labs have had to close so that social distancing can be observed. But sort of besides the challenge of that, which is something that I will come to in a few minutes, do you have a sense of how our view of disease has changed sort of broadly and how that might actually impact your work?
That is a very interesting question. Every time I speak about Leishmaniasis to somebody who hasn't heard about this disease, and you say, is it in South Africa? And you say no, and they're like, okay, so why work with it, right? If there's one thing that COVID-19 has taught us, researchers and the global community, is that disease does not know geographical borders. And just because the disease is not here, COVID-19 started in Wuhan, and here it is even in South Africa. Leishmania is endemic in East and West Africa, yet we don't have it here. So you just need a few infections. There's no geographical location that's free or that's safe from any disease. That's the main thing it has taught me and the community, my colleagues, etc., even my students.
Yes, of course. So, back to that point though, how did you manage with the social distancing sort of shutdown that was required in South Africa with your lab?
Essentially, it was a very difficult time, and it still is a very difficult time. Because of using mouse models, we have to maintain the breeding of the animals, and we have to maintain what we would call the growth of the parasites in the animals, which means the students, based on the Veterinary Council regulations in South Africa, have to check on the animals and their parasites daily, which was not possible during lockdown. So essentially, we had to stop lab work. We had to get our parasites, freeze them down so that when we are able to come back, we're able to basically have to start again from scratch. So we would have to restart these models that we've been working on over this time.
Of course.
So it's been very challenging thus far.
Yes.
Because of the nature of the work.
Yes. So now let me ask you a potentially tough question to answer, but I'll ask it anyway, Ramona, which is that based on your work, your extensive work in infectious disease, work with Leishmania, and what you've seen with the pandemic in terms of COVID-19, if you had a magic wand and could change sort of broadly how people learn about science, what kind of education is provided, even in the public realm as well, is there something in particular that you would want? So, for example, the public to understand about science and the doing of science in the face of what you've seen happen, both with your work, but also more recently with COVID-19?
Very good, very interesting question. And I think it comes down to one of the main things is to build an awareness of something before it hits. Everybody knows HIV, like I said, malaria, TB, but there's actually so many. And because of what we know, we try to tend to group everything into the same, but that they're not. Each disease has its own way of functioning and causing the disease. So if there's one thing I would love to do is to build awareness. And I don't think we should be building awareness only at the adult. We need to start at the school level so that the younger children themselves understand, and this builds on. The way to do this, I think, is what people relate to these days, social media, but social media from more reputable sources or in a language that they can relate to, not the high scientific terms that they're not going to engage with, but in the simplest sense to build that awareness. Talking to actual scientists, actual laboratory technicians, not Dr. Google, because the internet can give you lots of information. But things like LabXchange, things like university sites, etc., which provides this information from reputable sources that people can relate to in a language that they can relate to.
So when you're not in the lab, when you're not reading scientific papers, when you're not having discussions about science with colleagues, what do you do? What do you enjoy doing?
Two large extremes. So I've tried my hand at boxing when I want to let off a bit of steam. Yes, but that's quite intermittent. But what I do more regularly is dancing. I'm training in Indian classical dancing for the past 8 years in Kathak. It's a classical dance form of Northern India. I haven't been able to attend classes over the last few weeks because of the lockdown, of course. But this is where I invest my time when I want to get away from the lab.
Well, sounds like a fantastic way to blow off steam and to sort of get your mind into a different state.
Absolutely. Right? At some point, except when I need to write dance exams and I have teaching and research and all of that going on at the same time. So then I wonder.
Dance exams?
Yes.
Did I hear that correctly? What's a dance exam?
So Kathak is like a syllabus. You have 5 different levels that you need to get through to graduate as a Kathak dancer. So with each level, there's a practical exam, a written exam that you need to go through, etc., to show that you've acclimatized with syllabus.
Wow. So it's like, there are a lot of exams that we all have to go through in science.
Yes.
And even when you're relaxing, you're doing something that has exams.
Exactly. I remember when I finished my honors degree and I thought, oh, this was the last of exams. I'm not going to miss this. And here I am.
Right. So, you work with model organisms. Is it fair to say that you have a favorite? And is it the one that you work with?
I would say so, to some extent. However, when I was in my PhD, I attended a workshop in Switzerland at the University of Lausanne with the WHO and an international research training group on Leishmaniasis. And at this meeting, I met researchers from Leishmania endemic areas. And within their presentations, they shared with us what the disease looks like at a patient level. Mm-hmm. And it was a complete awakening from what I was seeing in my model organism compared to what is actually going on with patients. So as much as I appreciate and love working with the model organism, at some point I would love to be able to work with patient samples and actual patients who are afflicted with this disease, because that was an eye-opener.
Ah, fantastic. Yes, yes, I can understand that. As a cell biologist myself, I did make a transition to human cells. I never worked with whole organisms in a significant way, but it is true that a particular interest in human application does lead you towards trying to work with human materials, but also with patients.
Ultimately. But also with patients. Absolutely. There's that personal factor that you don't see in the laboratory when you're working with the model organism. It's not just about finding a gene that could be targeted as a host therapy. It's a person's life that is severely afflicted by this.
Yes, 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 of course. So one thing that I think a lot of our viewers and listeners think about is the fact that the world is changing so fast, right? It used to be the case that, you would get a degree and have one job, right? And that your job would sort of stay somewhat the same and you might retire in a job that was very similar to the one you started with. As you know, we're all in a world now where people change careers, or even in the career they're in, you change direction, you change focus so frequently. And that happens a lot in science, which I know it's something you've experienced. So what folks often ask or wonder about is when you feel a change coming on, Ramona, how do you get ready for it? What do you do? How do you pick up what you need to, let's say, have a new direction in your lab?
I've actually have experienced that both career and personal wise. So I moved between my master's and my PhD, moved university, moved disease model, and moved city. And so that was a challenge. Moving city and moving away from what it is is also a personal challenge at the same time.
Yes.
And then I think the third one came when I moved from being a student to an academic staff member, when now you have to run your own research lab and you have to supervise students and you're responsible for their degrees, etc. So the way I do it, it's challenging and it's stressful at the time, but something I've always maintained even as a child is think ahead. Where do you want to get? What do you want to get from this? And where do you want to be after this? Do your research, do your homework, find out as much information. I think fear comes from not knowing. So if you try to get as much information as possible, you're comfortable or you're confident that you can get through it. It's the same with research. The research is not in the book. We have to read and we have to find out and we have to trial and error. You should be not afraid to trial and error. Some of the greatest findings came from error, and we should take comfort in that. And this is what I feel, both on the personal front and on the professional front.
Yes, yes. Fantastic advice and a real insight to share with our viewers and listeners. So Ramona, let me close then with one last question, which is, if you weren't a scientist, if you weren't a world-class researcher, what would you be?
You're probably going to laugh, and everybody who's going to listen to this interview is going to laugh. But like I just said, my passion, my non-academic interests lie in dancing and theater. So I'd probably be in Broadway.
Ah, fantastic.
As an actress or a dancer. If I had to choose. So yeah.
Okay. That's completely fair. And it gives insight into sort of another thread of passion that drives you.
Absolutely. Yeah. I think we all need a balance. There has to be something, like you said, that lets off steam. There's two sides to us. There's the academic side, and there's something else that we enjoy, but we see the greater good in what it is we do. So yeah.
Okay, that's fantastic. Well, Ramona, thank you so much for joining us. on the Exchange. And I know that your lab is starting to open up, I think, bit by bit. And I know that you will continue to make significant contributions to how we understand human disease. So thank you so much for joining us.
Thank you so much and the team for having me. It was a pleasure speaking to you all, and I wish you well in this endeavor.
All right, thank you.